In the summer of 2010, a team of scientists in Germany announced the existence of an entirely new species of ancient human. They gave it a name. They described where it had lived. They even estimated how it had spread across an entire continent. And they did all of this without a single complete bone. No skull, no skeleton, not even a full skull fragment large enough to hold in your hand. The entire discovery rested on something almost absurdly small, a chip of finger bone barely the size of a pebble, and a scrap of tooth, both pulled from the floor of a Siberian cave. What those two fragments contained wasn't a shape scientists could measure with calipers. It was a code, a sequence of chemical letters buried inside ancient cells that, when finally read correctly, revealed a species of human that no one on Earth knew had ever existed. This is the story of how a bone smaller than a fingernail clipping quietly rewrote the human family tree, and how that discovery forced scientists to admit something uncomfortable, that we had been walking past the evidence of a lost branch of humanity for years without knowing it was there. If stories like this one, the moments where a single piece of evidence quietly rewrites everything we thought we understood about our own origins, are the kind of thing that keeps you curious, go ahead and subscribe, because that's exactly what this channel is built around. Now let's walk through exactly how this discovery unfolded, from the cave, where it began, through the two separate scientific papers that slowly revealed what the researchers were actually looking at, to the strange twist buried in the data that nobody expected to find, and finally, to what this discovery opened up for an entire generation of genetic research that followed it. To understand how strange this moment really was, you have to picture the world of paleoanthropology just before it happened. For more than a century, discovering a new species of ancient human meant one thing above all else: finding physical remains substantial enough to study. A skull to measure brow ridges and cranial capacity. A jaw to compare tooth wear and shape. A pelvis to determine how something walked. This was a science built on bones you could hold, measure, photograph, and argue about in academic journals for decades. Every major discovery in the field, from Java Man to the first Neanderthal skeleton pulled out of a German quarry in 1856, followed the same basic formula: find enough physical structure to build a convincing case that you were looking at something genuinely different from modern humans. That original Neanderthal find, and even that modest amount of material, sparked decades of fierce argument among scientists about whether they were looking at a diseased modern human, an ancient soldier, or something else entirely. It took years of additional discoveries across Europe: more skulls, more skeletons, more careful comparative anatomy. Before the scientific community broadly accepted that Neanderthals represented a genuinely distinct, extinct form of human. That was the standard of proof the field had operated under for well over a century. A standard built entirely around the physical shape of bone. Genetics, when it eventually entered the picture in the late 20th century, was initially used only to confirm or refine conclusions that fossils had already suggested, never to make the initial discovery on its own. Nobody had ever announced a brand new species of ancient human based purely on a genetic sequence, without a recognizable skull or skeleton backing it up. That template was about to be broken entirely, and it would happen in a cave most people outside a narrow circle of Russian archaeologists had never heard of. Then, in 2008, a team of Russian archaeologists working in a limestone cave in the Altai Mountains of southern Siberia, a site called Denisova cave that had already been excavated on and off for decades, pulled a tiny fragment of bone out of a layer of sediment. It came from a finger, and it was so small and so unremarkable looking that under any other circumstances it might have been logged, bagged, and left in storage for years before anyone gave it a second thought. But this particular site had a reputation. Neanderthal tools and remains had already turned up in the region, and the cave itself had produced other fossils that hinted at a longer, more complicated history than anyone had fully mapped out. So the fragment was sent to a laboratory in Leipzig, Germany, to a research group led by a scientist named Svante Pääbo, who had spent much of his career trying to prove that genuine, readable DNA could be pulled out of remains far older than most of his colleagues believed was possible. What happened next unfolded in two separate acts, and the gap between them is part of what makes this story so remarkable. In the spring of 2010, Pääbo's team published an initial result based on the mitochondrial DNA extracted from that finger bone. Mitochondrial DNA being a small, separate piece of genetic material passed down only from the mother, easier to recover from ancient samples because cells contain many more copies of it than they do of the much larger genome stored in the nucleus. The mitochondrial sequence was strange. It didn't match modern humans. It didn't match Neanderthals either, whose own genome the same lab had been racing to complete around the same time. It looked like something else, something that appeared to have split off from the rest of the human family tree long before Neanderthals and modern humans even separated from each other. At that point, the scientists knew they were looking at something unusual, but mitochondrial DNA alone wasn't enough to draw firm conclusions. It's a small sliver of the total genetic picture, and on its own it couldn't tell them whether this was a genuinely new species, a strange regional variant, or some kind of statistical anomaly. So the real test became sequencing the nuclear genome, the full genetic library stored inside the chromosomes, a task that in 2008 or 2009 would have been a technical long shot, attempted on a bone fragment this degraded and this old. By December of that same year, the second paper landed, and this time there was no ambiguity left. Researchers had managed to sequence roughly 70% of the nuclear genome from that same finger bone, achieving what geneticists call about 1.9-fold coverage, meaning on average each part of the genome had been read and cross-checked nearly twice over, enough to compare it directly against both the modern human genome and the newly completed Neanderthal genome the same team had published only months earlier. The comparison was unambiguous. This individual was not a Neanderthal. It was not a modern human. It was something that shared a common ancestor with Neanderthals further back in time, a sister lineage that had split off and gone its own way, surviving somewhere across Asia for tens of thousands of years, while almost no trace of it showed up anywhere in the existing fossil record. The scientists gave this newly identified population a name drawn directly from the cave where the evidence had been found. They called them Denisovans. And here's the detail that made the entire discovery feel almost like science fiction to the researchers involved. When they later analyzed a separate tooth found in a different layer of the same cave, its mitochondrial DNA matched the finger bone almost precisely, confirming that both fragments came from the same population, even though the tooth belonged to an entirely different individual. But that tooth had something the finger bone didn't: actual physical shape. Actual morphology scientists could examine directly. And its shape didn't look particularly close to a Neanderthal or a modern human at all. If anything, its proportions echoed something more like far older, more primitive members of the human lineage, structures reminiscent of species like Homo habilis or Homo erectus, human relatives that lived over 1 million years earlier. In other words, the physical evidence and the genetic evidence were telling two different stories at first glance, one suggesting an ancient, almost primitive-looking hominin, the other placing this population much closer, genetically, to Neanderthals and modern humans in the family tree. That apparent contradiction became one of the strange puzzles the discovery left behind, a reminder that shape and genetic relatedness don't always move in lockstep, and that appearances, even in bone, can be deceiving in ways scientists are still working through today. What really turned this from an interesting academic paper into a discovery that reshaped an entire field was what came next, when researchers compared the Denisovan genome against a wide sample of modern human populations from around the world. The vast majority showed no meaningful trace of Denisovan ancestry at all, but when the comparison reached populations from Melanesia, particularly people from Papua New Guinea, the numbers jumped dramatically. Somewhere between 4 and 6% of the genetic material in some of these modern populations traced directly back to Denisovans. Not a rounding error, not statistical noise, a real, measurable, repeatable signal embedded in the DNA of living people thousands of miles away from the Siberian cave where the original bone fragment had been found. That single fact turned the story upside down. This wasn't simply the discovery of an extinct species that had vanished without a trace. It was the discovery of an extinct species that had interbred with the ancestors of modern humans, spread its genetic material across an enormous stretch of the ancient world, and quietly survived, not as bones in the ground, but as living code inside people who had no idea their ancestors had ever crossed paths with anyone besides other modern humans. Think about the scale of what that implies. For Denisovan DNA to show up at meaningful levels in populations from islands in the Pacific, while Denisova Cave itself sits deep in continental Siberia, thousands of miles inland, this population must have ranged across a huge portion of Asia at some point in the past, encountering the ancestors of the people who eventually migrated toward and settled the Pacific Islands. Researchers looking at this pattern in 2010 openly speculated that Denisovans might have been far more widespread across the continent than the single Siberian cave where their remains happened to survive would ever suggest on its own. It's a sobering thought for anyone who assumes the fossil record gives us a complete picture of the past. Here was direct proof that an entire ancient human population could range across a continent for tens of thousands of years and leave behind, as far as anyone has found to this day, essentially one cave's worth of degraded fragments, while its genetic legacy quietly persisted in living people on the other side of the world. The discovery also forced a reckoning with a much older and more stubborn scientific assumption, the idea that modern humans expanded out of Africa and simply replaced every other archaic human population they encountered, with little to no interbreeding along the way. That theory, sometimes called the strict replacement model, had dominated thinking in paleoanthropology for much of the 20th century. The Neanderthal genome, published by this same research group only months before the Denisovan paper, had already begun cracking that theory open by showing that people with ancestry outside Africa carry a small but real percentage of Neanderthal DNA, evidence of interbreeding. Rather than pure replacement, the Denisovan discovery didn't just support that emerging picture, it multiplied its complexity. Now, there wasn't just one archaic population that modern humans had encountered and mixed with. There were at least two in different parts of the world, leaving different genetic fingerprints in different modern populations, a signal that would keep expanding in the years afterward as more ancient genomes were sequenced and more subtle traces of long-vanished human relatives kept turning up in living DNA. There's another layer to this story that rarely makes it into the short version, and it's a little unsettling once you sit with it. To extract usable DNA from ancient bone, researchers often have to physically destroy part of the specimen, grinding a section of it into powder, to chemically isolate the genetic material trapped inside. In the case of the original Denisovan finger bone, a portion of the fossil was ground up during this process, with the rest of the physical bone returned to the archaeologists who had excavated it. For years afterward, that piece of history briefly became something of a minor mystery in its own right. As techniques advanced, researchers wanted to re-examine the remaining fragment more closely using newer imaging methods, only to find that pinning down its exact condition and appearance took real detective work nearly a decade later, since so little of the original fragment survived and photographic documentation from the initial excavation was limited. When new images and analysis of what remained finally surfaced years afterward, they revealed something almost quietly poignant, that this individual, likely a young girl based on later genetic analysis, had unusually slender finger bones, delicate in a way that added one more small human detail to a story that had, up until that point, existed. Almost entirely as abstract genetic data on a computer screen rather than as a physical, tangible person. That tension between data and person sits at the heart of why this discovery felt so different from every human origin story that came before it. Every previous discovery of a new hominin species had been built around a physical presence you could stand in front of, a skull in a museum case, a full skeleton laid out on a table, something the public could look at and immediately understand as evidence. The Denisovans arrived in the world stripped of nearly all of that. There was no skull to put in a glass case, no striking photograph of a full skeleton to run in newspapers, just a technical paper full of genetic comparisons, population statistics, and percentages of shared ancestry, describing an entire vanished branch of humanity that most people would never be able to picture in their minds, because there was almost nothing to picture. And yet, the scientific impact was just as significant, arguably more significant in some ways, because it proved that an entire species could be identified, described, and placed correctly on the human family tree using nothing but genetic evidence, without requiring the kind of physical remains that had been considered essential for the entire previous history of the field. That shift in method didn't stay contained to just this one discovery either. Once researchers demonstrated that a new hominin species could be identified through DNA alone, even from a fragment too small and too nondescript to ever earn its own display case, it opened the door to an entirely new way of searching for lost human relatives. Instead of waiting for archaeologists to stumble across a sufficiently complete skeleton, geneticists could now go back through drawers of previously excavated, previously ignored bone fragments sitting in museum collections around the world, fragments too small or too ordinary looking to have ever been flagged as significant, and test them for exactly this kind of hidden genetic surprise. Some of those fragments have already delivered second and third confirmations of Denisovan presence at other sites, including bone and dental remains later identified on the Tibetan Plateau using techniques that could recover information from proteins even when DNA itself had degraded past the point of recovery. Each new fragment added a little more geography, a little more evidence that this population had genuinely spread across a huge stretch of ancient Asia, rather than existing as some isolated curiosity confined to one Siberian valley. It's worth pausing here to be precise about exactly what was established by this original discovery and what remained genuinely uncertain in the years that followed, because it would be easy to overstate either side. What the 2010 papers conclusively demonstrated was that the Denisova cave fossils represented a genetically distinct population, closely related to, but clearly separate from Neanderthals, that this population had interbred with the ancestors of some modern human groups, and that its genetic legacy persisted at meaningful levels in living Melanesian populations specifically. What remained far less certain at the time, and in some cases still involves active debate today, includes exactly how widely Denisovans ranged across Asia, how physically distinct they truly looked compared to Neanderthals and modern humans given the scarcity of complete skeletal remains, and precisely when and where the interbreeding events that left Denisovan DNA in modern Melanesian, and later confirmed in other Southeast Asian and East Asian populations, actually took place. Responsible reporting on this discovery has always had to hold both of these things at once, genuine, rigorously, confirmed scientific fact sitting right alongside open questions that required years of additional fossil hunting and genetic comparison to even begin answering. What makes the story worth returning to more than a decade later isn't just the scientific technique, remarkable as it was. It's what the discovery quietly implies about how much of the human past might still be hiding in plain sight. Not buried under some remote mountain waiting for a dramatic excavation, but sitting in drawers and storage boxes and museums that have already been dug through. Once, fragments already collected, already cataloged, already sitting there for years, simply waiting for someone to apply the right technique and ask the right question. The Denisovans weren't found because someone went looking for a new species of ancient human. They were found because a small, easily overlooked bone fragment happened to end up in the hands of a lab willing to test whether genuine ancient DNA could survive well enough to be read at all. And when it turned out that it could, it revealed a population that had been hiding not underground, but inside the living genetic code of people scattered across the modern world, waiting patiently for the science to finally catch up enough to notice they were there all along. There's a reason this discovery still gets cited as one of the pivotal moments in the entire history of genetics and human origins research, but the story of Denisova Cave wasn't finished delivering surprises. Nearly a decade after the original announcement, researchers went back through the cave's collection of unremarkable, previously unstudied bone fragments and picked out a small, splintered piece of long bone cataloged simply as Denisova 11. Almost nobody expected it to be significant. It was exactly the kind of fragmentee that had sat in storage for years, too small and too nondescript to warrant urgent attention. When scientists finally sequenced its DNA, the results stopped the research. Community cold. Roughly half of this individual's genome matched Neanderthal ancestry. The other half matched Denisovan ancestry in proportions so clean and so evenly split that there was really only one explanation left standing. This wasn't someone with mixed ancestry several generations back, diluted and blended the way most admixture in modern populations appears in the genetic record. This was a direct, first-generation child of a Neanderthal mother and a Denisovan father, a young girl whose parents belonged to two different distinct branches of the human family tree and who had, at some point, found themselves together in or near the very same cave. Researchers nicknamed her Denny, and her genome remains one of the single most striking pieces of direct evidence ever recovered, showing that these separate archaic human populations weren't just distantly aware of each other's existence somewhere out in the vastness of ancient Asia. They were close enough, at least occasionally, to meet and to have children. That discovery reframed the entire Denisovan story a second time. It wasn't just that this vanished population had, at some point in deep history, contributed a small percentage of DNA to the ancestors of modern Melanesians through interbreeding that happened, who knows how many generations back. It was that the different archaic human groups roaming Ice Age Eurasia were, at least sometimes, encountering each other directly, likely more often than the fossil record could ever hope to show, given how rarely bones this fragile and this old survive at all. If a single cave, excavated only in fits and starts over several decades, happened to preserve direct physical proof of one such encounter, in the form of an actual mixed ancestry child, it forces an uncomfortable statistical question. How many other encounters like this happened across the enormous stretch OF Eurasia these populations occupied, leaving behind no trace at all because the remains simply never survived, were never found, or were sitting in a museum drawer somewhere, still waiting for someone to test them? That question has only grown more pressing as genetic research has expanded beyond Denisova Cave entirely. In the years since 2010, researchers studying modern genomes from populations across Africa have identified faint, ghost-like traces of interbreeding with archaic human populations that have never been matched to any known fossil at all. Groups geneticists can only describe indirectly through the statistical shadow they left behind in living DNA, populations we may never find physical remains of, whose existence we only know because their genetic fingerprint survived in people alive today. In other words, the method that first identified the Denisovans, reading ancient code rather than waiting for a skeleton, has since revealed hints of still more lost branches of humanity that may never be confirmed by an actual fossil discovery at all. Some of these ghost lineages may forever remain exactly that, a statistical echo with no bones ever to associate it with, no cave, no tooth, nothing, except the quiet mathematical certainty that a group of extinct people who once lived somewhere on this planet left a small trace of themselves in the blood of populations who have no memory that they were ever there. There's a reason this discovery still gets cited as one of the pivotal moments in the entire history of genetics and human origins. Research. It didn't just add one more name to a list of extinct hominin species, it fundamentally changed what counts as evidence in this field, proving that a molecule, properly read, can tell you things about the deep human past that bones alone never could. Before 2010, the story of human evolution was written primarily in stone tools and skeletal remains. After 2010, it was being co-written by something far smaller and far stranger, invisible strands of code hidden inside cells, patiently waiting in caves, in permafrost, in museum drawers, for scientists finally equipped to read what they had been quietly recording all along. The ripple effects of that shift are still spreading through the field today. Museum collections that once seemed fully exhausted, cataloged decades ago and considered scientifically settled, are now treated as potential goldmines, since nobody knows which unremarkable drawer might contain the next fragment capable of rewriting a chapter of human history. Techniques for extracting genetic material from increasingly degraded, increasingly ancient samples have improved so quickly that specimens once considered hopeless are now being revisited with methods that didn't exist when they were first excavated. And the underlying assumption that once anchored the entire field, that you need a face, a skull, something visibly and dramatically human to declare a new species, has been permanently retired. A signature hidden in a chemical code, invisible to the eye and undetectable by any measuring instrument except a sequencer, turned out to be enough all along. There's also something worth sitting with about what this discovery reveals concerning the limits of what any single field of science can tell us on its own. Paleontology gave researchers the cave, the sediment layers, the archaeological context needed to date these fragments in the first place. Genetics gave them the code that identified an entirely new population, but neither discipline alone could have delivered the full picture. It took both working in concert, along with steadily improving laboratory techniques capable of coaxing readable information out of material that had been degrading in cold cave sediment for tens of thousands of years, to arrive at a conclusion this significant. That kind of collaboration across disciplines, paleontology, genetics, chemistry, statistics, has become the default model for how major discoveries in human origins research get made today, a direct legacy of what started with one overlooked finger bone from a cave in southern Siberia. And perhaps the most important lesson embedded in all of this has nothing to do with ancient Siberia at all, and everything to do with how much confidence we should place in the completeness of any story we think we already understand. For well over a century, the accepted history of human evolution felt reasonably settled. A handful of known species, a handful of known migrations, a story that mostly made sense with the fossils already in hand. Then a single overlooked bone fragment, ground down for testing almost as an afterthought, revealed that an entire branch of humanity had been hiding in plain sight the whole time, not buried deep underground waiting to be excavated, but scattered quietly through the genomes of living people who never knew their ancestors had a story this strange to tell. It's worth remembering, too, that this entire discovery almost didn't happen at all. If that particular fragment had been slightly smaller, slightly more degraded, or simply overlooked during excavation, the way countless similarly unremarkable fossil chips have been overlooked at other sites throughout the history of archaeology, the Denisovans might still be nothing more than an invisible statistical signal, a faint genetic echo in modern Melanesian populations that scientists could measure but never explain, an unsolved anomaly rather than a named, documented branch of the human family tree. That razor-thin margin between discovery and permanent obscurity is easy to forget. Once a finding becomes textbook knowledge, cited in papers and documentaries as though it were always destined to be found, it wasn't. It survived by chance in a cold, stable cave environment that happened to preserve exactly what was needed, at exactly the moment scientific technique had finally caught up enough to read it. Carry your story well. It has come a very long way. Sources consulted: Krauss, J. et al. The complete mitochondrial DNA genome of an unknown hominin from southern Siberia. Nature 464, 894, 897, 2010. Reich, D. et al. Genetic history of an archaic hominin group from Denisova cave in Siberia. Nature 468, 1053, 1060, 2010. Max Planck Institute for Evolutionary Anthropology Press Materials. Neither Neanderthal nor modern human. 2010, Green, R.E. et al. A draft sequence of the Neanderthal genome. Science 328, 710, 722, 2010. Bennett, E. A. et al. Morphology of the Denisovan phalanx closer to modern humans than to Neanderthals. Science Advances 5. Galen O. Asmero, 2019. Harvard Medical School news coverage of the Reich et al. 2010 nuclear genome analysis.